Alright, so let's try to look at enzymes and biochemical reactions. Alright, so I've gotten these questions from different past papers we are revising under enzymes. So you need to understand what an enzyme is, as well as the characteristics of enzymes and the effect of those characteristics of enzymes, as well as the chemical reaction or the effect of enzymes. So an enzyme is just defined as a biological catalyst or a biological catalyst that speed up a lot of a chemical reaction. Now, when you include the fact that these enzymes speed up of the chemical reaction, meaning you're talking about a catalyst, because you know to say a catalyst is anything that can speed up a lot of a chemical reaction, and you know to say that a catalyst is the one that lowers the activation energy. So from there, we have this question which is saying figure 2.0 shows a graph showing the effect of temperature on an enzyme catalyzed reaction. So we have this graph here. So one of the factors that affect the rate of enzyme reaction is temperature. So you're going to find that a human body operates on the normal temperature of 37 degrees Celsius, or we can say 35 degrees to 37 degrees Celsius. This is the normal temperature of the human body. Now exceeding this temperature, meaning to say that even the work of the enzyme is going to be affected, in that the enzymes, they are going to undergo denaturalization. So when enzymes are subjected to higher temperature greater than 35, or we can say greater than 37 degrees Celsius, you're going to find that they are going to undergo denaturalization. Now when I say denaturalization, I just mean that the shape of an enzyme is just going to change permanently without being reversed. Okay, now one thing that you need to understand here is that, as you can see here, the graph is starting from somewhere 0, increasing in the rate of the chemical reaction. So here we have the rate of the chemical reaction. So as you increase the temperature, you're going to find that even the rate of the chemical reaction is going to increase. So when the temperature is low, even the enzymes are going to be inactive, meaning they're not going to be active. So as the temperature is increasing, even the rate of the chemical reaction is going to increase up to this point here. So this is somewhere 37 to 35, 35 to 37 here. So this one, you can say this is 37 degrees Celsius. Now as you can see here, this point we call it as the optimum. Now specifically, optimum. So specifically, this term optimum, since you're talking about the temperature, so it's going to be the optimum temperature. Now the optimum differs when you're talking about the optimum of the pH, meaning you're going to say the optimum pH. Now we're talking about temperature, meaning to say it's going to be the optimum temperature. So what is the optimum temperature? Optimum temperature is the point or the temperature at which the enzymes work are the best. Meaning to say, exceeding this temperature, you're going to find that the enzymes are going to undergo denaturalization. So beyond this curve here, so going down here, the enzymes are undergoing denaturalization up to 30, let me just say 60 degrees Celsius. So up to 60 degrees Celsius, that is where we're going to find that there will be complete denaturalization. So complete denaturalization is occurring at this point here. That is one thing that you need to understand. So the first question is saying, what is an enzyme? So to define an enzyme, we are going to say it is a biological catalyst. As simple as that. Biological catalyst. Okay. Then when you talk of a catalyst is anything that can speed up the rate of a chemical reaction. Now, the work of enzymes, they can speed up or they can slow the rate of they can speed up or they can slow the rate of the chemical reaction depending on how the reaction is proceeding. Then the next question is saying, what is the optimum temperature of the enzyme reaction shown in the graph? So as you can see here, we can just imagine that this is 37 to 36, but approximately that is 37 degrees Celsius, because we notice that is the normal temperature of the human body. So it has to be 37 degrees Celsius. Then at what temperature in figure 2.0 is the enzyme completely denatured? So like I said, it is 60 degrees Celsius. So denaturalization was occurring, as you can see from this curve here. it was just occurring until this point where we are having 60 degrees Celsius. Then give a reason for your answer. So we can just say this is because that is where the curve is ending. This is because, okay, that is where the curve is ending. The curve is doing what? It is ending. Like that. Then the next one is saying, which is Roman number 1, question C. Explain why it is important to measure the temperature, the body temperature, when a person is sick. So it is very, very important to measure the temperature when the person is sick to know the optimum temperature of the enzymes. So that is to know the temperature suitable for the enzymes, right? That is to know the temperature suitable for enzyme activities, Then apart from that one, it is very important to measure the temperature for diagnosis. So what I mean about diagnosis, you're going to find that when the temperature is high, you're going to suspect that someone is having a certain disease. Like for example, if someone is having a fever, meaning to say you're going to say this one is having malaria or other diseases. So you can say for diagnosis. Okay. Then apart from that one, Roman number 2 is saying propose 2 ways the body temperature of a sick person can be brought down to normal. That is by giving a person aspirin. So by giving a sick person aspirin. So aspirin is a medicine that lowers the body temperature to normal. Apart from that one, you can also put a wet cloth on the forehead of a person to lower the body temperature. That is putting a wet cloth. So when I say wet cloth, a cloth which is having moisture, or that you have put in water or soak in water. So a wet cloth, by putting a wet cloth, okay, just a cloth on the forehead. Right. So those are the answers for that question. Let's see the next questions. Alright, so question 2 is saying figure 2 shows the stages involved in an enzyme catalyzed reaction inside cells of the living body. So we have G and H binding on this side, and we have structure I. After binding on this side, they are coming together. It's more like they are building up from this broken one, the smaller particles, into the larger molecules. So from here, the question is saying from stage 1 in figure 2, identify the enzyme and the substrate. So a substrate is made up of 2 parts. We have this part here, which is called the allosteric site. The allosteric site. So this allosteric site is just the other part of an enzyme. And then these 2 parts here, they are called the active site. Okay, so this part is called the active site. So the active site is just a place where now the substrate binds. So we have these 2, G and H. These 2, they are called substrates. Okay, so we have to identify which one is an enzyme. So we're just going to say I, because I is pointing at this one here, which is an enzyme. Then the substrate, we can say G and H. These 2, they are substrate. Then which stage shows the enzyme substrate complex? So E, when you have enzyme substrate complex, that is where now you are going to have the substrate binding to the active site of an enzyme. So as you can see here, these 2 substrates are binded to the active site of an enzyme. So we have this enzyme as well as the substrate. So we can say it is stage 2. That is where we are having the enzyme substrate complex. Then other questions. We have to label the active site of the enzyme, which I've already labeled, which is which are these two parts. So you can identify all these parts here, they are active sites also. Then from that question also have this one, which is saying what type of metabolic reaction is catalyzed by the enzymes. So one thing that you need to understand is that when you talk of metabolism is just the sum total of all chemical reaction that occurs inside the living body. And these metabolisms, they can be anabolism as well as catabolism. Anabolism is where now small molecules are being brought together or are being built to form large molecules, or they come together to form a large molecules. And what facilitate that are enzymes. So in short, enzymes, they can be used to build large molecules from smaller molecules. They can also be used to build up or to break down large molecules into smaller molecules. So from here, they are asking us the type of metabolic reaction that catalyzed by the enzyme in figure 2. So let's see. So we have these two small pieces which we are calling them as the substrate. And this substrate, they are smaller they are small quantities. Then after the reaction, they are coming together, meaning to say we are coming up with a big molecule. So these are small molecules and we are coming up with big molecules, meaning to say this is anabolism. But the reverse is true, where if this is the large molecule, if it binds to the active site of an enzyme, then you are forming these two, meaning to say that is catabolism. So that is one thing that you need to know. So this is anabolism, building up anabolic or anabolism. So anabolic or anabolism, you are building up large molecules from small molecules. And then the question is saying, the second one is, which is saying, name a region in the human body where such a metabolic reaction in Roman number 1, which is question C, Roman number 1 above, can occur. So you can talk of the muscles as well as in the liver. So you can say in muscles as well as in the liver. Now, what is the reason why we're choosing the muscles as well as in the liver? You know, to say that when insulin is secreted by the pancreas, this one here, it converts what? It converts glucose into liver glycogen or muscle glycogen. So muscle glycogen is going to be stored in muscles and the liver glycogen is going to be stored in the liver. So the conversion is like this. We know to say that glucose is a monosaccharide. So once it is converted to glycogen, so glycogen is an example of a polysaccharide. So we are building the small amount, or we are building a large amount from the small amount, which is we are building the polysaccharide from the monosaccharide. So monosaccharide, we can just say they are FGG. So FGG means we have fructose, galactose as well as glucose. So these, they can be used to build up the polysaccharides like starch, glycogen as well as cellulose. They can be built from the monosaccharides. So that is the explanation here. We are changing glucose into glycogen, which is a polysaccharide. Then Roman number 3 is saying, state the general term used to refer to enzymes that work inside cells. So the enzymes that work inside cells, they are called intracellular enzymes. So let me just write it properly. So they are called intracellular enzymes. So intra means inside. So intracellular enzymes. Intracellular enzymes. And those that works and those that operates outside the cells, they are called extracellular enzymes. So that is one thing that you need to know here. Then question D is saying, describe two industrial application of enzymes. So enzymes, they are used in daily. Okay, so let me say, daily. Can like diary here. So daily industries. Okay, so they are used in daily industries. Why do enzymes being used in daily industries? So there are some meats where which are hard to eat for the babies, and those meats, they can only be softened by using the enzymes, right? So these enzymes are used for, hello, just a minute, just a minute, just a minute, just a minute. I'm recording a video, just a minute. Then from there, we can also use enzymes in leather tanning. So when you talk of leather tanning, so leather tending or animal tenderizing, so meat tenderizing, tenderizing meat or meat tenderizing, as well as leather tanning. So when you talk of leather tanning, this is where now the enzymes are used to soften the animal skin, so that they are able to make shoes from the animal skin, as well as other products like bags and other things. All right, so let's see the last questions. So of course here we have the combination of movement of substances across the cell membrane as well as enzymes. So we have figure 2.1 shows a table of substances absorbed by plants from the soil. So we have oxygen, nitrate, potassium, as well as water. So from figure 2.1, identify the substances or substance which enters the plant by osmosis, active transport, as well as diffusion. So when you talk of osmosis, water enters the root air cells by osmosis. So we can say water here. Water enters the root air cell by osmosis. Then when you talk of active transport, in this case here, we're talking of the ions. So the ions, they use the ion channels for them to enter the membranes. All right, so as we can see here, we have the nitrates as well as the potassium. These, they are ions. So we can say the nitrate, these, they use the ion channels. So the nitrates and the potassium. Okay. Then from there now, we have diffusion. As we all know that during distillation, oxygen enters the alveoli into the, them say oxygen enters or passes through the alveolar or alveolus by diffusion into the bloodstream. So here we are going to say oxygen. Then the last question, which is saying figure 2.2 is a graph showing the effect of pH on the rate of enzyme controlled reactions N and O. So we have reaction N as well as reaction O. So as you can see, the reaction N is operating under acidic. So this is the pH scale, which runs from 0 up to 14. So the pH from 0 up to somewhere here 6.9 is acidic, and then the pH of 7 is called neutral. Then the pH from 7.9, or we can just say 8 up to 14, this is called alkalinity pH. So N is operating under acidic and O is operating under alkaline or basic pH. Then when you talk of X and Y, these X and Y, they are representing optimum. Now specifically, since we are talking about one of the factors or one of the characteristics that affect the enzyme reaction, which is the pH. So meaning to say X and Z, it is going to be the optimum pH. So that's the difference between optimum pH and the optimum temperature. So meaning to say, when you talk of optimum temperature, it's just the, let me say, when you talk of optimum pH, it's just a pH at which enzymes work are the best. So we have Roman number 1. What term is given to the part labeled X and Y in the graph? So we have X and Y. So X and Y is referred to the optimum pH. As simple as that. Don't say optimum pH, don't say optimum temperature, because here we are dealing with the pH. So that is one thing that you need to know. Then we have this one, which is saying which reaction is under acidic condition. So the one which is under acidic condition is reaction N. So I can just say reaction N. Then reaction O is under alkalinity or basic reaction. And then which reaction could only be for a protein food, and give a reason for your answer. So you know to say that the digestion of proteins takes place in the stomach, where pepsinogen is converted into pepsin or into active form, which is pepsinogen. And that pepsinogen or pepsin catalyzes the reaction of proteins reactions. So you are going to say it's reaction N, the one which is under-acidic. So reaction N. And then the reason for our answer, this is because we know to say that enzymes that catalyze proteins work well under acidic. So it's just going to say protein enzymes work well under acidic. Protein enzymes work well under acidic. As simple as that. So we are done for the revision under enzymes. Thank you very much for watching, and remember to subscribe for more videos like this.